Okay, so we're going to start now. Hi, everyone. Thank you for attending our Early Career Members Network live webinar series. My name is Daniela Ramirez. I'm a postdoc researcher from UNCuyo University and CONICET fellow from Argentina. I'm also an EMN committee member, and today myself, along with Diana Pinto, from the Instituto Superior da Engenharia do Porto in Portugal, and also an EMN member. We will be co-hosting the 15th session of the EMN live webinar series from the Metabolomics Society. But before introducing today's we wanted to remind you this session will be recorded and posted on the Metabolomics Society website. In case you haven't done it yet, we encourage you to join our social media network via X or Twitter, or following our LinkedIn page for updates and featured content. You can also send us an email to make suggestions or ask for further information. In this opportunity, we have the pleasure of presenting Dr. Álvaro Fernández-Ochoa's research work entitled Untargeted Metabolomics Approaches in Nutritional Intervention Studies, uncovering insights into bioactive compounds. Dr. Álvaro Fernández-Ochoa earned his chemistry degree at the University of La Rioja in Spain in 2014, receiving the Extraordinary End of Studies Award. He completed his PhD in chemistry in 2019 in the University of Granada, also in Spain, achieving summa cum laude and the Extraordinary Doctorate Award. He conducted then postdoctoral research at the Max Delbrück Center for Molecular Medicine in Berlin, Germany, before returning to the University of Granada, where he works as a professor, and he combines teaching with research there. His work focuses on developing untargeted LC-MS metabolomics methods for clinical studies and nutritional interventions, with emphasis on the bioavailability and metabolism of bioactive phenolic compounds. He is also a member of the Metabolomics Society, the Quality Assurance and Quality Control Consortium, and the Spanish Society of Metabolomics. He was a prior member of the EMN, the Metabolomics Society, and he's currently a member at the Education and Training Committee of the Metabolomics Society. He has collaborated in teaching among various universities, including Latin American institutions, the University of Rio de Janeiro, Universidad de Costa Rica, or Universidad Nacional de Tucumán in Argentina. So we really appreciate Dr. Fernández-Ochoa's presence today, and we hope you enjoyed this session. You can start now, Álvaro.
So let's see. Let me see if you can see my screen, Daniela or Diana. Is everything okay?
Yeah,
Okay,
everything's
great.
okay.
Great. So first of all, thank you very much, Daniela and Diana, for this kind introduction, and also for this invitation. For me, it's a really pleasure to have this opportunity to give a webinar. As Daniela introduced me, I was a EMN member from 2021 to 2023. So I was in that side 2 years ago. So and now it's a pleasure to me to have the pleasure to have here a talk. And I will try to do my best today. So also I prepared some slides about my background, but Daniela introduced me very well. So I am a chemist. I studied my bachelor degree in the University of La Rioja, which is located in the north of Spain. And then I moved to the south in 2015, almost 10 years ago, and where I did my master's students and also my PhD students in chemistry in this university. During that period, I went also during 3 months in Chalmers University in Sweden, supervised by Carl Brunius. That period was very challenging for me to learn a lot of data processing tools, how to process and target metabolomics data, and was for me one of my most important periods in my research which changed a lot my vision in metabolomics. So, and when I finished my PhD, also my postdoc was in Berlin in the Max Delbrück Center in the lab supervised by Jennifer Kirwan, and also these 2 years were also for me a lot of learning about metabolomics. And now I am back. I went back to in 2022 in the University of Granada again, and I am still here. In general, also I prepared this slide because as I said before, so I was a EMN member from that period. For me, was a very also beautiful experience for me to meet a lot of international people, doing a lot of things like these webinars, activities in the conference, many networking activities. So for all early career members who are attending these webinars, so I really recommend you to try to join this committee. Every year there are positions open to join. So for me it was a period who changed a lot my academic career. So I would recommend you for all of you. And I know that Daniela and Diana can you also tell you later some of the positive things that who have take care of or join this nice community, or join also the activities who are very well organized by this committee. So thanks a lot for being a speaker invited for this nice EMN committee. So in my career, in my research career, so we know that untargeted or metabolomics applications that there can be in a lot of fields, but mostly, so from my PhD until now, so I mainly focus on developing and applying untargeted metabolomic approaches. That is something that I wanted to talk today. And I have the chance or the opportunity to work in 2 different types of applications. I have collaborated in projects more related to diseases, to look for biomarkers for better prediction of disease or the prognosis. So I had the chance to work with systematic autoimmune diseases, also with cancer in 2 important projects during my research. But also, on the other hand, that is something that is the topic of today's webinar. So I have the chance also to apply these untargeted metabolomics methodologies to the study of bioactive compounds, especially performing some dietary intervention studies. That is something that I want to explain today in different ways, like some acute intervention studies to study the absorption, the viability, the pharmacokinetics or metabolisms of these bioactive compounds, but also some longitudinal studies to determine the impact of the intake of these bioactive compounds in the endogenous metabolism. So this is the topic of today's talk. So before moving to the main topic of how we can apply untargeted metabolomics to study further these bioactive compounds, so let's start. Which are these bioactive compounds? So basically, they are general compounds who are generally present in natural sources like plants, especially, and they can show some beneficial effects on the human health. They can be classified like 2 types, like nutrients that we generally need to intake them daily, but also non-nutrient compounds. And in this family, we have the phytochemicals. And we have different families in which we can cover some different type of phytochemicals, as we can see in the slide. But mostly in my introduction, in my topic of today, also we are going to focus on phenolic compounds, because it's one of the most important family of phytochemicals, because there are thousands of phenolic compounds reported in the literature, as I am going to show in the next slide. And generally, so what are the interests of these compounds or these phytochemicals or bioactive compounds present in vegetal sources? Is because if we can say a strong evidence that they can have important beneficial effects on human health, for prevent diseases, for treatments, for whatever. So we can develop some functional foods, for example, some nutraceuticals that we can sell to the community. And in fact, you can look for right now, you can see many functional foods or bioactive extract, where you can buy in in some shops based on bioactive strat, for example, olive leaf. That is one of the most sources or vegetal sources that we are working in our research. So you can go to Google, look for, I want to buy a supplement based on olive leaf strat, and you can find hundreds of this type of supplements. So the way is, or the question is there still enough evidence or scientific evidence of the intake of these strats or these compounds daily can support or can benefit the health in general? So it's something that we need to invest more in research to know better the mechanisms, and for that we can use some of the advances in untargeted metabolomics or metabolomics in general to have a better knowledge or better evidence of the mechanism or the beneficial effects of the intake of these supplements or these compounds in general. So I wanted to focus today in phenolic compounds because there are hundreds. Right now, there are more than 10,000 structures reported. So it's a family that there are a lot of molecules of this family. And there are a lot of structural diversity. So you can imagine in 10,000 structures, you can have different types. Also, there are a classification of phenolic compounds depending on different chemical structures. And also, depending on the vegetal source, you can find different phenolic families, or rich in flavonoids, or rich in other families, or specific compounds. But in general, there is like a general message that these compounds that they are present in many botanical sources, vegetal sources, and there are differences in the structure. So there are in general same bioactive properties. So similar bioactive properties reported for most of these compounds. So in general, you can see phenolic compounds in general, they usually have antioxidant properties, anti-inflammatory, antimicrobial, or some of these that they are listed in this slide. So one of the questions is how these bioactive properties that usually are reported for this type of compounds have been usually tested. So generally, this has been discovered by a traditional bottom-up approach. That is something that I want to show in this slide. So in the first step, so it's trying to look for a strat rich in bioactive compounds, selecting a plant or a strat of a plant or part of the plant, and trying to have a strat rich in bioactive compounds. Then trying to study if this strat that we can obtain for the plants, they show in some in vitro study some positive results in bioactive test. If they look positive results, then we move to, okay, let's try to know the composition of this strat, which phenolic compounds are behind the strat, and then you can start using analytical techniques to discover the compounds. And then we move to a further step using maybe animal models to have more evidence about these strats or these compounds, or move more further to human models to show more evidence. And if finally we have positive results in all the models, maybe we can end formulating of a new supplement or functional food or that we can sell to the population. So also I want to say something here is like in this traditional bottom-up approach, maybe starting looking for which compounds are present in a botanical strat, we are performing a phytochemical characterization of a strat. So we start working with metabolomic approaches, although we have only one sample, and we want to know which compounds are in that sample. So most of some PhD students who are starting in my lab usually say, I want to do metabolomics, blah, blah, blah. But generally in my research lab, our students start using phytochemical characterization of a strat. And as I want to show you in the next slides, is you are starting working with some of the typical important steps of the untargeted workflow steps, okay? So as I said before, so this is like a general way to discover or to generate evidence of the properties of the bioactive compounds, like having strats rich in these compounds using conventional techniques or advanced extraction techniques like microwave extraction or another techniques. This is not the topic of today, but there are a lot of research about how we can use different extraction techniques to have better recoveries or efficiencies of the strats rich in these compounds. Then the bioactive tests, using in vitro animal models, human models, if it's possible. And one of the important steps is also the characterization. And as I said before, if in this step, we are starting doing some metabolomic strategies. So for people maybe who are coming here more from the topic of the bioactive compounds, of the phytochemicals, and maybe they are not so familiar with the metabolomics field. So generally metabolomics is, by definition, is trying to analyze low molecules, low molecular weight molecules, like below 1,500 Daltons. So all the chemicals who have a molecular weight below this value, 1,500, they are in the field of metabolomics. And phytochemicals, like all the samples that I put before, like phenolic compounds in generally, they are metabolites by this definition. So we are starting, if we are analyzing a botanical strat, we are starting doing some metabolomic analysis. And in generally, metabolomics is classified in targeted and untargeted. So targeted analysis in general is focused on some metabolites predefined before the analysis. So we know I want to analyze if this strat or this sample have different vitamins or different phenolic compounds that I know that I have evidence that they can be there. So I focus or I develop an analytical method to analyze them. And in a targeted, we have the opposite. We don't have the predefinition of the compounds that we want to analyze in the strat. So we are going to try to cover as much as we can in the analysis in the LCMS run or GCMS run or NMR or the analytical technique to try to cover as much as we can. So in the field of phytochemical characterization, that it can be the first moment to start doing some metabolomic analysis. So we can have this type of application. So we can run some targeted analysis when we want to quantify some bioactive compounds in some bioactive strats. We want to know the concentration of some compounds that we know that they are there. So we are doing some targeted metabolomics analysis. And on the other side, when we don't know the composition of the strat and we analyze them to know it, we are doing some untargeted metabolomic analysis. Although we don't have hundreds of samples that we are going to show later in the samples of the intervention studies. So I like this scheme that was published in a manuscript of Galard Ayala, that it's a good definition of the difference of targeted and untargeted. What is better? So depends on the application. So if we know the compounds and we want to quantify, it's better to go for a targeted analysis because it's going to be more selective and we are going to have more sensitivity. We can analyze metabolites that are in concentrations below than if we perform an untargeted analysis. And the main difference is one of the main decision is which instrument should I use for an untargeted analysis or targeted analysis. So you can say here, so for an untargeted analysis, so the data acquisition if we use mass spectrometry, has to be done generally by the high resolution instrument, like a time of flight, an Orbitrap. And for example, if we are going to do a targeted analysis to detect compounds or quantify better compounds in a sample, we are in a low concentration. So the best way is low resolution, like triple quads, for example, or Q-traps. Okay. So this is something that a good definition of the difference and also which MS analyzer should I use for each application. So as I say, so maybe we are going to see later. So maybe this can be the general workflow for an untargeted metabolomic strategies, like especially when we are talking about biofluids, looking for biomarkers. So we need to start with the study design, with the volunteer recruitment. We are going to show the full workflow states later in an example of an acute intervention study. But before moving that, so as I want to focus in this type of phytochemical characterization samples, in which we have only one sample, and I think it's a good start to get familiar with some of these steps. And in that workflow, for doing a phytochemical characterization, so first of all, we need to, of course, prepare the sample, extract the metabolites, and get like a sample in a vial to get analyzed by the instrument that we want to use. Of course, the data acquisition. The sample has to be especially if we talk about MS spectrometry, that is the general technique that I am going to talk in this talk. So we need to select the data acquisition method, especially if we are going to do phytochemical characterization, as I said before in the previous slide. So we should use an untargeted metabolomic approach because we don't know which compounds are present in the strat. And for that, we should use a high resolution mass spectrometry, like a cutoff, an orbitrap. And then, so we need to move to the next step. So we need to get familiar with the data preprocessing. Of course, we only have one sample, but not only one. Please, if we are going to characterize bioactive compounds in plant samples or strats, use blanks, especially to remove contaminants or artifacts that can be in the chromatogram and they are not related to your strat. So please use blanks and also analyze your sample and then get familiar with the signal, try to do the picking, to deconvolute them, and of course to have some important MS data carried by high resolution mass spectrometry. Now we will see to get and to try to identify them using different sources that I am going to explain in the next slide. So for the phytochemical characterization, so we have different techniques. Which technique is better to use depends on the compound that you want to analyze. So I think 3 or 4 years ago, we published this review in which you can see different application of techniques, so GCMS or LCMS. But if you have, for example, LCMS, you can use different columns, different modes, positive, negative, reverse phase, helix. So also one key message that I want to show, so analyze all the metabolites in one single run is almost impossible because there are metabolites that they are, for example, you use a reverse phase, you have very, very polar compounds, they are not going to be separated by the column, or depending on the ion source model, you are running, for example, in positive mode or negative mode, so they are potential metabolites that maybe they are not very well ionized depending on the ionization mode. Of course, NMR is something that I don't have the expertise of, but is the most important technique to have a very good compound elucidation. If you have different isomers, if you want to know exactly which isomer is, so if you are able to isolate this compound and analyze by NMR, so it's the typical technique that you can predict the isomer based on the NMR result. So that is a technique very useful for that. And also likely, so that the techniques and are still progressing and we have new advance. And now we have, for example, also a new separation dimension apart from the chromatography that is also ion mobility spectrometry. That these instruments are now in use, people are using, and of course, this dimension, when it's coupled to, for example, with chromatography plus ion mobility spectrometry plus mass spectrometry, so it's very powerful technique because the ion mobility spectrometry can separate metabolites which were not separated by the chromatography because they are based on another phenomenon. They are separated depending on an electric field. And then we have more data to use for also I know that now in the databases, they are reporting more these collision cross-sectional values that they are very useful to annotate, to identify, for example, isomers of different compounds. Repeating again, then the previous slide. So as I say, we are running untargeted metabolomics workflow, so we should use a high resolution mass spectrometry. So like a cutoff, Q orbital. So why? Because we have very good advantage in when you use, we have very good quality data that they are very useful to predict structures. First of all, we have a mass SAT, very accurate. Below, we should have an error below 5-10 ppm using this type of instruments. So and also the isotopic distribution with also a very good mass accuracy. So with this data, usually we can have a very good prediction of the molecular formula. But of course, with a one molecular formula, we can have many isomers with this formula. So we need more data. So for that, it's better to use a MSMS analyzer like a QTOF or a QORBITRAP because we can perform some fragmentation spectra. So we can isolate some precursors that we want to identify, annotate. We can apply some collision energy to have the fragmentation spectra, and then use this fragmentation spectra to predict better the chemical structure. For example, here is an example of a QTOF. So for example, when we have this type of analyzer, we can work in two types of modes. So for example, in the MS1 mode, when we talk about full scan, so it means that the quadrupole is not filtering, it's living from a huge range of a mass to check ratio. Like for example, if we are going to work in a full scan range, for example, from 50 Daltons to 1,200 or 1,500. So all the ions which have a mass to check ratio in this range is going to the time of flight. And then we acquire all the signals. But for example, in this example, we have the 166. That is something that we want to annotate. And for that, we want to obtain the fragmentation spectra. So then we can design a MSMS acquisition method. Also, the instruments or the general instrument that we can have now, in general, we can automatize this type of methods, especially doing some data-dependent acquisition methods or data-independent acquisition methods. My general advice, I had the opportunity to work with both, and if you want to have a better spectra, it's better to work, in my humble opinion, with data-dependent acquisition, because we can isolate the precursor. If we are doing data-independent acquisition, we are not isolating the precursor in the quadrupole. We are fragmenting everything from the range from 50 to 1,000. So, and then we need to doing a extra step in the data processing to try to separate the fragmentation spectra coming from potential two coeluting compounds. So for that, if you are, for example, doing some phytochemical characterization samples, so I recommend you to perform some data-dependent acquisition. And then we have the fragmentation spectra that I said before. We can using many tools, especially right now, one of the better things of our metabolomics field is we have a lot of open source tools that everyone can use. They are free. So like all of these tools that I am putting in this slide, they are totally free. So if you are not aware of them, so you can start using them whenever you want. So then one of the things, so when we have the masses at the isotope distribution and the fragmentation spectra, we can compare with databases. There are also tools for searching in different databases like CEU Mass Mediator, mass banks. There are also software for prediction. I recommend a lot Sirius also for doing molecular networking tools in which we can also annotate with comparison with different databases. For example, there is a very good tool like is GMPS. If you want to learn a lot of for Sirius, GMPS, there are also a lot of tutorials in YouTube. So the people who develop this tool, they are very, very kind, and they are providing a lot of tutorials, many things to try to learn in a very easy way. So I strongly recommend it. And also, for example, if you are working in this field of phytochemicals and you have the chance to isolate or have standards of some phenolic compounds or phytochemical compounds, and then you can perform some MSMS data of these isolated compounds. So also I recommend you to contribute to the expansion of these databases by sharing the spectra and putting the repositories, so all the community can get benefit of it. And one important thing that I want also to highlight here is how to report. And I also say it in this talk, maybe sometimes identification, but we need to be careful with that. Because with all the information, also doing the comparison with our data, with the databases, we need to be careful because we don't we cannot be 100% sure that this compound is 100%, although we have the analytical standard, and we analyze it, and we compare our data with the analytical standard run with the same instrument, with method, and we see, okay, the retention time of the component I want to identify with the retention time of a standard match, also the MS data, the fragmentation spectra, and then we can use the word identification when we are reporting and we discuss the results in the manuscript. But if we are in the level 2, level 3, we are using only the MS1 or the MSMS data and only comparing with these sources like Sirius, GMPS, or databases, comparing spectra from. Please never use the 10 identification. Use better the 10 annotation or tentatively identify or tentatively characterize it, because we cannot be 100% sure that is that component. Maybe we are 90%, 95%, but it's not 100%. So be careful and use this level. It was defined in 2007, and you can see I checked it yesterday when I was preparing this presentation, and right now there are more than this paper have more than 4,500 citations. So it's a very, very guideline that is followed by a lot of research. Okay, so now we introduce a little bit about the bioactive compounds, the phenolic compounds, some of the ways to identify or annotate compounds that it can be applied to phytochemicals compounds, or these methodologies using MS spectra, MSMS fragmentation, and compare with databases. It can be also applied to annotate compounds in biological samples, as I am going to show later. But what else do we know about phenolic compounds? So we know that once they are we intake the phenolic compounds, they suffer reactions before going to the bloodstream. So, for example, in the small intestine, there can be phase 1, phase 2 metabolism reaction. Like, for example, I put here one sample for the quercetin. They can be sulfated, glucuronidased, methylated. So we know, we have evidence, these reactions, they were reported years ago, and we know that when these metabolites are intaken, so they are transformed, they are suffering chemical reaction. And then also the reaction can be in the gut, in the liver, and they are in the metabolism, so they can be transformed through the metabolism. So, and how these biology or metabolism studies have been usually performed. So to check the biology or to see how these bioactive compounds are suffering reactions once they are intaken. So generally, the best way to analyze them is perform to an acute nutritional intervention study. So what do we mean with an acute? So basically, these studies can be done in animal models or also in humans. So basically, we took several biological samples, mainly blood, urine, feces, through a short time, like 24 hours to 48. And we took several, for example, blood samples at different times to see which compounds we have in different samples after the intake. The intake usually takes time 0. And after that, the samples are taken. But also, if we are planning to perform one study like this, we should also consider to take one sample before the intake to compare, okay? So 2 years ago, so we also do like a review about how people usually perform this study. And we see, of course, so the analyses are performed by by Metabolomics analysis, but we saw in this review that it's not frequently used untargeted metabolomics approaches to look for these viability metabolites or to discover more about the metabolism. So for that, we are proposing to use some of this untargeted workflow, and now we are going to move step by step to trying to apply an untargeted methodology to study the viability or the metabolism of bioactive compounds. So until now, so we are going to start with the first step, as you can see, is the study design. So the main objective is to apply untargeted approaches to acute nutritional intervention study. But before Before showing the results or the methodology that we applied, I am going to show maybe the premises that that we already talk about that. So first of all, so we know that there are numerous reported phenolic compounds in the literature. We saw also that there are a lot of structural diversity among the compounds present in different plant sources. In general, source rich in phenolic compounds are characterized by presenting similar bioactive properties. So although there are different plants with different specific phenolic compounds in their composition, at the end, they have similar bioactive properties. Also, I saw in the previous slide that we know the metabolization reaction that these phenolic compounds can suffer. And we also detected that untargeted methodologies have not been frequently used to study the viability or the metabolism of these compounds. So for that, we had our questions or hypotheses to answer in this study applying untargeted studies. So first of all, which compounds are responsible for the beneficial effects that can be reported for these type of families during years? The original ones, which are characterized in the strats, or the metabolized ones, once they suffer the reactions. And of course, they are in the organism. And also, could there be common circulating metabolites from different sources responsible for the common effects? So for that, we propose, instead of using this traditional bottom approach that I commented before, to change the way. So let's start, instead of starting with an in vitro assay, preparing strats. So let's start, we know a lot of evidence, a lot of many strats. So let's start doing an acute dietary intervention study in humans with some of these supplements or strats that have been well studied previously. And then let's try to apply here untargeted metabolomics to look for the metabolites which are in the biological samples. And then once we know the metabolites, let's study more about these metabolites, these circulating metabolites, doing some in vitro, in vivo assays in the further steps. So then the objectives of the study that I want to show today is, first of all, develop this step top-down approach based on untargeted metabolomics with experimental samples, mainly to explore the viability and metabolism of phenolic compounds from bioactive strats. Also, we want to look for common circulating metabolites coming from a strat with different composition but similar properties, and study the bioactive properties of the common circulating metabolite that we want to analyze them. So for that, we select different sources. So here we have Psyllium marianum, Lipia citridora, Hibiscus aldarifa, Theobroma cacao, or Olea europea. So we selected 5 bioactive strats to run this study. Why we select this? Because they have different, mostly completely different chemical composition, but all of them are rich in phenolic compounds, but rich in different families of phenolic compounds. And in generally, the 5 of them look similar bioactive properties. So as I said, the first step that we wanted to perform is, of course, preparing the strat, the supplements that was performed by a company in Spain, NATAC, and then perform the acute intervention state. So for that, of course, when you are going to perform an acute intervention or an intervention study in humans, so there are a lot of bureaucratic paperwork, of course, to have the permission of an ethics committee. So this requires a lot of time, a lot of work, and also it has to be planned in advance, the study. So also I'm going to show later one example of one of our current study of longitudinal study, and I can say that there is a lot of work before starting the study, especially for the paperwork, the ethics committee, also the recruitment. You need to recruit. So also when you are going to recruit, you need to define very well the inclusion and exclusion criteria of the volunteers. And then, for example, for this study, we selected 48 healthy volunteers recruited for the intervention trial. These 48 were divided in 6 groups. So remember that we have 5 supplements, but also we include a placebo group. So mainly, so each group have 8 volunteers. And the design of the study was like each volunteer came one day to, especially the intervention study of this study was done in the Universidad Miguel Hernández de Elche in Spain. And there, so the volunteers were there to one day, first of all, they bring the urine sample, and one blood sample was taken before the intake of the supplement. And then they took the supplement, and we started to took different blood sample at different times, 0.5, 1 hour, 2 hours, blah, blah. And also urine samples that were collected in different ranges to be analyzed later. Next, so once we have the samples, okay, I put here one tip. Like when you are performing this type of study with humans, there can be many people involved in the sample collection. You need nurses to perform the for example, the blood collection. So I strongly recommend you to have SOPs, like very well detailed protocols, simple, clear for the sample collection that everyone can follow in the same way. Depending on maybe one day there are one people working in this intervention, and other people in other day. Once the samples are taken, so they first of all they have to be very well stored for a metabolomics assay. So store at -80 is a very well recommended practice. In that study, we had plasma samples and urine samples. So if you have plasma sample and you want to analyze by, for example, in this case, an LCMS platform. So one of the main steps that you need or we need to do in the sample preparation is the protein precipitation, because the proteins can collapse the columns for the metabolomics analysis. So these protein precipitation steps usually are performed adding organic solvent like methanol, ethanol, acetonitrile. That is the main step that you need to take in consideration when you are planning to perform some untargeted or metabolomics analysis using LCMS in plasma samples. For urine sample, we need to take also another point into consideration, is that the urine samples depends a lot of the hydration status of the person. If one person drinks a lot of water, the urine of that person is going to be very diluted. And if we have another volunteer who don't drink in that time water or a lot, her or his sample is going to be less diluted. So it's something that we need to consider in any study in which we are analyzing urine samples. So we can have different types of urine, and there are also reported different normalization methods to correct this effect in the urine, depending on the level of water that was drank by a person. So there are methods of normalization method that can be applied after data acquisition, or before data acquisition. If I need to recommend one, I would recommend to perform a normalization method before data acquisition, because we can avoid ion suppression, or we can reduce the ion suppression effects in the ion source. Because if we don't do that, we can analyze high concentration samples that we are going to have more risk of ion suppression that in a low concentration sample. So it's better to normalize all the samples before the analysis, because all of them are going to be more or less at the same level. For them, the ion suppression effects are going to be more compensated between samples. And between you can normalize urine using, for example, the creatinine. It can be frequently used for many researches, but also other parameters like osmolality or specific gravity. In general, the three methods can be used, but for example, if you are using one study in some population in which you are researching a specific disease, creatinine can be risky, because creatinine is one metabolite, and if you are looking for biomarkers related to one disease, maybe that the creatinine can be a biomarker. So if you are normalizing, considering the creatinine, be careful. But for example, in one study, if you are using urine sample from a healthy population, it usually can be considered, because there are studies that evaluate that the creatinine level is generally related to this factor of the hydration. So that in our study, the plasma samples, as I said, we prepare doing the protein precipitation step, in that case with methanol, ethanol, and we perform osmolality normalization before the data acquisition. Then another important thing when you perform metabolomics analysis is to take into consideration QC samples, because they have a lot of advantages. You can use them to optimize methodologies like the sample preparation, the data acquisition, also for data processing. You can have one sample of a QC that can be used for this type of objective. When we talk about pure QC samples, it's like we can prepare that sample for a specific study, like basically by mixing a small aliquot of every single sample. Another use of this QC, they are usually used to equilibrate the chromatography system at the beginning of the sequence, to control the analytical reproducibility through the sequence. So this QC should be analyzed multiple times through the sequence. Later, I am going to show one example of how to design a sequence. Also, although we can have a very well control of the reproducibility of the, usually in this type of LCMS analysis or GCMS, that the sequence are very long, usually we have drifts. But also if we are running a lot of samples, because our study have multiple samples, also we can have between batch effects, differences between batches. They are also very useful for data curation, to filter low quality signals. And also it's a very good sample to representative all the samples of the study to be selected for fermentation. One tip that I want, or one advice that I want to say here for acute nutritional study, like the sample that I'm going to show. Okay, I say like in generally the pool QC sample, they are usually based of a small aliquot of all the samples of the study. But for example, in this type of study in which we are very focused on look for exogenous metabolites, and we know that in the placebo group or also the time zero samples, these metabolites that we are going to discover, they are not going to be present. So for example, I recommend to exclude this placebo group or the time zero group to preparing the QC, because if in these samples they are not going to be these compounds, and if we use these samples to prepare the QC, we are going to we are diluting the important compounds in the QC if they are present in the rest of the samples. So then in our study, we perform the data acquisition. In this case, we use a cutoff from Agilent. We work in the reverse phase mode with a C18 column and in the negative ion mode, and combining MS1 full scan mode and data-dependent acquisition MSMS mode. Here you can see the parameters, but I want to focus more on the sequence design. So as I said before, it's important to run in any metabolomic study, or to take into consideration quality assurance and quality control approaches. So for example, before starting a sequence, and system suitability tests should be run before starting the sequence to check if the instrument is ready for purpose, before putting all the samples in or all your important samples in the rack. If imagine you have a problem in the column, you have a leak in the system, so you need to check everything, and when everything is checked, okay, then go to the fridge, take the sample, and let's put in the LCMS rack. We talk about the use of QC samples. As I said before, they can use for equilibration at the beginning, use multiple times, run multiple times through the sequence, also perform MSMS data acquisition analysis in these samples to have initial fermentation spectra of most of the important metabolites present in them. And also another important test that you can do with QCs is also perform like a QC dilution series, to run different QC at different dilution levels, because we should them to remove contaminants or non-linear signals. So that is something that I would recommend. Also use blanks, especially at the beginning and at the end of the sequence. And also, for example, in this type of study, in acute intervention study, it's important to randomize the samples in the batches, so don't put all the samples from the olive group or the placebo groups in the first batch and in the second batch. So it's important that the samples have to be randomized. But I will use here, or we use, for example, clustered randomized by subject. So it basically means that all the samples, for example, we have an example on the right. We have all the samples from the volunteer one of the Lipia group, and all the samples were analyzed together, and then a QC, and then another group, but with all the samples, because then we are minimizing the intra-subject technical variability. Then once we acquire the data, so we move to the data processing step. And here, so we usually perform some steps. I don't have enough time today to talk about every single step, but some of the most important is the peak picking, the alignment, normalization, peak grouping, data processing before statistics to put the data ready for the statistics. Also the data curation for filtering contaminants or low quality signals from the final data set. So as I say, I don't have enough time in this talk to talk about the specific details of how to optimize different steps, but something that I want to say to all the community here is the different tools that you can use to perform this data processing. Of course, generally every single vendor software have their vendor company. You have an instrument for Agilent, you should have a vendor software designed to perform some of these steps. But apart from the vendor software, there are a lot of open source tools. For example, some of the most popular ones, MSMine, MSDial, MetaAnalyst, Workflow Metabolomics. So you can, as I said at the beginning, likely we have many open source tools for the community that we can use. That is great for everyone. And for example, for MSMine, I know it's, for example, one of the tools that I usually use in my research. I know that the other tools, they are also great. But for example, for MSMine, there are many tutorials in YouTube, free to watch and very easy to learn. So if you are new in this field, please go to these tutorials. And one of the most important things, or the positive aspect, apart of many tools, many algorithms, many functions that this software have, is that they are very friendly. They are very easy to learn. They are, because apart from these tools that I am showing, also there are many packages, programs based on R in Python for specific steps that you want to perform in your data. For example, for peak picking, one of the most famous packages is XCMS. There are, for performing, for normalization, you have batch core. So for different, also for annotation, so there are many R packages. You can go to the literature if you are expert in R, or you can use them. So it's great. So moving back to our study, so how we process the data. So in that case, we use the MSMine software to perform the peak picking, alignment, and different steps. So we obtain a lot of features for plasma and urine. Then we use an R package that also I recommend, that is Notame, in which package we can use some data curation steps, mainly for example, to remove contaminants using the blanks, also to normalize the data, to correct the drifts, the bitching batch effects, signal clustering. And after performing these steps, so we obtain like more than 4,000 features in plasma and more than 5,000 features in urine. So once we perform all these steps, so then we move to the statistical analysis, in which statistical analysis we can use univariate or multivariate analysis to detect the important metabolites. For example, here in one of the preliminary results that we obtained, so we use some univariate analysis, like a volcano plot, to look for significant metabolites depending on the groups that we analyzed, comparing the groups versus the placebos. And we obtain these results using this criteria, like FDR below 0.05 and fold change higher than 2. But after that, in this type of studies, one of the advantages is that we have the placebo group, also we have the time zero group. So we can use these groups for additional filtering. So if we are looking for a metabolite that we know that we have the prediction that is coming from the strat, so we can use the placebo group for filtering. So if one of the significant metabolites are highly present in the placebo, we should filter for the annotation. And here we have one of our example after filtering, and we obtain these values. Then, as I say, we can use other statistical models to try to detect the significant metabolite related to the intake. For example, some multivariate to also the PCA. They are very useful also to check the data quality, to check how the QCs are grouped in the PCA. Also, we also use some cluster analysis like heat maps. I saw before samples of univariate analysis, also linear models with covariate adjustments. But as I said before, also a part of using the statistics, we have also some important samples that they can be used for filtering, like the placebos or the time zero samples. Here I saw some examples of the multivariate analysis, like the heat map. So the colors are allowed to the different groups, the different plants. So here we highlight, for example, the highly numbers of metabolites significant related to the intake, for example, detected in urine. All these group so are significant metabolites or significant signals that is clearly because they are highly abundant only in this column. And this color, as you see, allows to the OO group, that is olive oil, olive strat. And so we have many signals in urine in comparison to the plasma. And in fact, we are still working with the urine, processing the results of the urine sample that we are planning to report them soon. Here, for example, we have another cluster related to the significant metabolites or highly present only in the volunteers who intake the Silibum marianum strat. And here we have another example from the Lipia Citrodiora strat. And one of the positive things when we saw these results is that we have an overlap here between the olive and the Lipia strat. That is one of our objectives, that we wanted to look for these common metabolites coming from different botanical strats. Unfortunately, we were not able to find one common metabolite through the 5 strats, but we were able to find at least some common metabolites in plasma and urine also, only with olive and Lipia strats. So here we have an example of the volcano plots in which we have the numbers and we highlighted the common significant results between olive and Lipia, and also the result that I saw before in urine, but also we detected some of them in plasma. So we move to the annotation step. For that, I explained it in the introduction in the introduction of the presentation. We talk about the phytochemical, how to characterize the phytochemicals in one botanical strat. And then when we are here it's exactly the same. So we have some significant signals in the samples and we want to annotate. So we have now the information of the MS high resolution mass spectrometry, as I say, MS1, isotopic distribution, the MS fragmentation spectra, and we compare with, or we can use the comparison with all the tools that I saw at the beginning. And here, so the preliminary results showed that we were able to annotate, for example, 9 common metabolites in plasma and 15 in urine. That was data from a month ago, but we are still increasing the as I say, the results in urine, and we are increasing this number at the moment. But now I am putting here some examples, and I want to highlight that most of the significant metabolites, they are metabolized. So, or they are sulfated derivatives or metabolites who suffer glucuronization reactions. So, but we could not able to identify in the plasma or in the urine the original compounds present in the strats. So we know that at the end, in the plasma samples, in the urine samples, they are mostly detected the metabolized one instead of the original ones. And unfortunately, most of the research about the bioactive properties of the bioactive strats are based on the compounds present in the original strat instead of these metabolized compounds. That at the end, we saw it, or not only us, many researchers about who performed similar studies in another with another strat, there are evidence that then they are these metabolites in the organism. So we need to have more research about how are the role of these the derivatives in the organism or in relation to the bioactive properties. So here we have some examples of, I am highlighting some examples of these common metabolites, and something also very interesting in our results of this that we were able to annotate that, for example, here we have some common some of the common metabolites like the vanillic acid sulfate or homovanillic acid sulfate, and we know that these compounds that they were not very related to the original composition. We detected that they can come from the degradation of the hydroxytyrosol, that is one of the compounds, or from higher structure like oleuropein, which is present in the olive strat, or verbascorsid, that is present in the lipia strat. But one of the most interesting results is when we analyze it in which moment was the highest intensity in plasma sample. We detected that depending on the strat who the volunteers intake, the time in which the abundance or the intensity in plasma was higher was completely different between the people who intake the olive, that was at the beginning through the 1 hour, than the people who intake the lipia. So that was one very interesting. And it was not only in these two metabolites, it was in generally in most of them. Here you see the final result that we report recently in one manuscript, and we saw that the trends were completely different between the two strats. So we know that we were able to annotate one common metabolite between the two these olive and lipia strats in plasma, but we discover that depending on the original strat, the mechanisms are completely different because maybe they are coming for completely different higher compounds. So you're more interested in these results. So we recently published these two manuscripts like 2 months ago. So in Food and Function and in the Journal of Agricultural and Food Chemistry, you can see in one of them is more related to the development of the untargeted metabolomics applied to the study of this type of strats. And another is highlighting this role of these common metabolites that we were able to annotate between olive and lipia strats. So, and what's next? So, as we say, so now we have a picture of which metabolites are present in the biological samples, and we saw that most of them are metabolized, coming from the original ones. So now the next step in our proposal of the innovative top-down approach is trying to isolate them and try to study them in vitro or in vivo models. Especially the isolation can be done, or firstly, if the analytical standard we can buy in any commercial website or company, so it's perfect. So we can acquire them, but unfortunately, not all of these sulfate or glucuronidase derivatives are present they are present as analytical standards. So another way is try to isolate them by purification methods, or try to, for example, use synthetics to try to get them isolated to test them later in vitro, in vivo models in a further detail. So I want to show you very briefly that, for example, one of our PhD students in our lab, Maria del Carmen, went to California to perform a PhD stay, and we acquired some of these commercial standards like the vanillic acid sulfate, homovanillic acid sulfate, and others. So we are working now in that publication that we hope that can be reported very soon. And we test these isolated compounds in some pancreatic cancer cell lines using some cytotoxicity study or also Western blots. Here, there are some of these results for these two compounds, but others were also tested. For example, we saw, for the MTT test, that, for example, the toxicity between the vanillic acid sulfate was significantly different versus the vanillic acid So that was quite interesting. So for example, the vanillic acid can show a better evidence related to these lines. Also, they discover more about the role of in which with the Western blot analysis, in which this can be related with these proteins. And we have, as I said, working more in this data. Also, we have more metabolites which were tested, and we are still open for collaborations like this to get more knowledge about the role or the bioactive role of these derivatives that we know that at the end they are present in the biological samples. Also, I want to conclude this webinar also talking about new perspective, also talking about longitudinal intervention study. So we also, I saw examples of how an acute intervention study are done, but there are also longitudinal intervention study. The main aim of that is to determine the impact of the intake of the endogenous metabolites, of the intake of these phenolic compounds in the endogenous metabolites after a period of weeks or months. So it's basically we need to recruit some volunteers. They need to intake this supplement, extract, or isolated compound through weeks or months, and we should take different biological samples through before starting the intake, also after finishing the intake, and then compare these samples to see or to check the impact on the metabolism that the intake can have. So in that case, we had the EcoVelivo project that we run in our lab. So I want to say that this project, so I went back to Granada from Germany when I finished my postdoc in 2022, and in that moment the project was starting, and we were working a lot of time, as I said, in the bureaucratics to get the permission of the ethical committee. Then in the recruitment, also imagine that once we were able to recruit all the volunteers, there was a project that takes per volunteers like 5 months, so it's not so quick as an acute intervention study. So 2 years later, now we have the data. So it's a lot of time before starting the project until we have the data, and I hope we can have the results very soon. So that is the idea of the project. So in that project, we focus in an olive leaf supplements. We were able to recruit around 100 volunteers. They were split in 2 groups, and we performed 2 phases per volunteer. In one of them, the half then took the placebo. Then we established a washout period, and then they finished the second phase with the opposite supplement in the second phase. And in this project, so we want also to do metabolomics analysis, also using multiomics analysis in trying to integrate also microbiomics. So it's something also the new approaches, trying to integrate different omics to get more knowledge, interactions, and that is also. So as I say, so we have the data finally, at least of the plasma samples. Also, we have more samples that we are planning to analyze next year, but at least we have the data from the plasma sample. We are still working on it, and I hope to see you in Prague next year. And I hope that we can have and show you the preliminary results in that conference. But before ending the webinar, I want to also to say some general consideration if one day you are planning to run some of this study. So in this type of study, as I say, since the volunteer start the first day until the volunteer ends the study was around 5-6 months. So then when you are planning the recruitment, you should expect that a number of volunteers, they are not going to complete the full study because of different reasons. Because maybe they don't want to, maybe they have a problem, they move to another city, many things can happen. So for example, when you are planning a recruitment, you need to consider an additional number of volunteers than the numbers of you need for the statistics, because in 5 months it's very likely that a small percentage they are not going to finish, although they were recruited. Also, you need to organize informative meetings with them before starting the study. You need to provide very clear and simple information for the understanding. For example, you have here an example of the information that we provide to them. This study was run in Granada in Spain, so the information is Spanish, but we provide this information, this paper. In this paper, they have the important information. When they come, when do they need to come to the different visit, which samples do they need to bring, and all this type of information. And also, some of the things is that you need to follow up with volunteers throughout the study. Also, you need to be open every day to receive calls or message from them to maybe to try to answer them any question that they can to have, and also follow all the consideration that we talk about related to the acute nutritional intervention study, like in general, use QC, SOPs for the sample collection, and all the things that we consider. So this is the end of the webinar, and I think I spent a bit more time than 1 hour, but sorry. And some of the take message that I want to say is that there is a great opportunity to apply the latest advances in untargeted metabolomics to study more the role of the bioactive compounds. Consider the top-down approach that we propose or we present in this webinar. The acute intervention study using no-target approach help us to detect unexpected circulating metabolites coming from different sources, common metabolites as we saw. Also, we were able to predict or to see different mechanism related to one specific or single metabolite depending on the source. It's necessary to explore more the bioactive properties of these metabolized compounds that we are able to detect in biological samples after the intake, and also consider the longitudinal intervention study to determine the impact of the intake of bioactive compounds on the endogenous metabolism and to have more evidence about the effects of the intake of these compounds on the health. So before ending, I want to acknowledge especially my research group because they are the most important to show all these results today. Also the projects, the funding agencies, especially Junta de Andalucía, the government of Spain, the EcoBio project, also the collaborators especially for the results that I saw in this project, the Universidad Miguel Hernández and the UC Davis, and especially also the volunteers that without them, this research or this study will be totally impossible to run. And also I want to acknowledge all the people who are or were related to me in my research life. Especially I want to mention Carl Brunius and Jennifer Kirwan because they were very pioneers to me or very inspiring to me to learn a lot about metabolomics and good luck, but also all the society, all people, all collaborators that I have throughout the world, like also Daniela that is here. So it's all of them. So thanks thanks a lot. Thanks to the EMN again for inviting me to have the chance to present this webinar today.
Thank you, Álvaro, so much. Thank you for your enriching presentation. It was very clear and very detailed. It was like a class on metabolomics. Well, thank you for sharing your expertise on how to address bioactive compounds, characterization, and nutritional interventions through metabolomics. I think it was really, really detailed. We have 4 questions. Maybe Diana, you wanna
Yes, I can start.
Okay.
So, with the questions. So, Álvaro, the first question is, please discuss normalization here in pre-processing.
Okay, it's an important topic. It's something that you need to consider, especially for untargeted metabolomics is more challenging because, for example, in targeted analysis, you can acquire internal standards, and then you can normalize your chromatograms based on internal standards. But for untargeted metabolomics, that is very challenging because we cannot have one representative or 1 or 10 or 20 internal standard that can be used for all the metabolome or all the hundreds, thousands of signals that we are able to detect, because every single metabolite can have a different trend in the mesh in this case. So for that, as I said before, it's very important to use QCs in your runs because you can monitorize them very well. You can use them as a trend, and you can normalize your data or the drifts. If we're talking about normalizing the drifts or the between batch effects that we can have. So having the same samples run multiple times through the same batch or multiple batches, so it's very useful. And you can find many methods in the literature based on them. And for my side, is the method that I will recommend you, especially in my side. That, for example, when I started to work in metabolomics, so it's the main big problem that I had in my first project. It was related to normalization, because I ran my first PCAs and I saw my QCs in line, like related to the injection order. And then I need to learn how to normalize it. And in my personal experience, I need to move to Sweden with the Carl Bruni's professor, and he showed me, for example, one of his packets in our packets that he developed, that is the batch core, and it was based on QCs. And for me, is the one of the best ways to normalize. So you have any comments, or you have never used it, or any question, or how to start using? So write me an email. I think now in the slide you can see my networks and also my mail, and I can share with you scripts or tools to normalize them. But this is related to the normalization of the drifts or the analytical variability in your data. That maybe I understand the question like that. But also remember, or in the presentation, I also show the sample of the urine. That also we need to normalize the effect, for example, related to the water or the hydration status. That we can do before, as I recommend, before the analysis, using, especially I would recommend osmolarity, or you can use also another metrics like creatinine if you are using with urine.
Yeah, so thank you. The next question is, hello, I have a question about omics tools. Is there any tool that focuses on food omic analysis? I am looking for tools that can analyze all the components of a food at once.
All the components of a food. So because there are many components in a food, like proteins, big molecules. So you need different methods, different approaches, different. If you are trying, for example, how now here we are focused on the metabolites, on the small compounds. So in this type of analysis, we are not detecting the proteins or the other big compounds of the present in the food. So it's very challenging to have one single method or analysis to I think it's impossible, from my opinion. You need to combine different techniques that they are detecting different things based on different methods. Unfortunately, hopefully in the future.
Thank you, Álvaro. So the next question is, could you kindly share it with us? Thank you for the excellent presentation. Could you kindly share it with us? I also have a question. What tools do you prefer for data visualization?
So of course, I think the webinar was recorded, so I think it's going to be
Yeah.
uploaded in the Metabolomics Society website, so totally free for everyone. So
Yeah.
for data visualization, so I can imagine that is for statistics, or for example, to visualize the PCA or the results. So as I said before, so some of the tools that is or I like it. So for example, if you need to visualize the chromatograms and you don't have, for example, a vendor software in your computer, so you can use, for example, MS-MIND. You can see the chromatograms and everything. If you want to, for example, to visualize the statistics, maybe you are not very familiar with R-code, or one of the best or easy way to visualize and have also very quick results is Meta-Analyst. It's a web server. You can upload your data and you can do multiple statistical methods without a lot of, you don't need a lot of knowledge, and they are very fast, and you can have a very quick visualization results. But also in R, in Python, you can optimize your graphics or everything you want. But Meta-Analysis, I think, is a very good tool nowadays to visualize the data.
So the next question, thank you for sharing this presentation. Do you study the impact of the whole food on human health?
So in our studies, or in the longitudinal study that we are running, so basically we are studying the impact of the strats based on the in this case in an olive leaf extract, rich especially in this type of compounds, phenolic compounds. So basically the idea is so we are not studying the impact of one of these isolated compounds that we detected, because I think to run it, we need to have a very strong evidence that this specific compound is the responsible of the effects to go or to move to a longitudinal study using that. And for that, we are performing some in vitro study to starting to get evidence. And maybe one day, if we want combine in vitro with animals, maybe one day we can use it for using it to get the evidence in humans. But right now we are trying to use olive extracts, because as I said at the beginning of the presentation, you can go to Google, try to buy a olive leaf supplement, and you can buy it. And they recommend, because they can have antioxidant properties, many of these bioactive properties, but we need more scientific evidence of the effects. And for that, we are performing this study in the EcoBiolivo project, in which we want to discover what are the effects of the intake of this type of supplements through 2 months in the metabolism.
Thank you. So the next question, what kind of QCs, kind of internal standards with different concentration?
So as for untargeted metabolomics, I say, so using internal standards is challenging, because they cannot, so they are not going to be representative for all the metabolites that you are analyzing in your system. For that, my recommendation is to use or prepare a QC, like a pure QC, like using all your samples in your study and trying to get aliquots for the same similar aliquots for every single sample, and put in one vial or in one falcon all these aliquots together, and have like a pure QC pool sample representative for all of them. But as I say, for example, in this type of acute intervention study, maybe you can exclude some of them if you are looking for exogenous molecules. Don't use the placebo sample, but it's an exception of this type of specific study. But in a general way, in an untargeted study in general, so use all your samples and prepare your pool QC. If you don't have, or you are running, for example, a study in which you don't have enough amount of sample in your, because you have small aliquots and you cannot prepare or use enough volume to prepare the QC, also another way is to buy it, buy reference samples, buy commercial samples. You can buy plasma commercial sample in some websites. It could be another option, but my recommendation should be a pure QC.
Thank you, Álvaro. So one more question. Thanks for the brilliant presentation. My question is how to go from the step of filtering and normalization when you have thousands of features to the next steps where you reduce a lot of the number of features. I mean, how to reduce the number and obtain the good features. Thanks.
So it's basically to define very well the filtering criteria, because as also as I saw in the samples of the presentation at the beginning, we had detected like 20,000 features, but maybe 5,000 were related to contaminant or artifacts that they were also detected in the blanks. So the blanks can be one of the first exclusion criteria to filter variables. Also, using, for example, detection rate exclusion filtering criteria. For example, it can be that you are analyzing, in that case, we have 48 volunteers, that every single volunteer have their diet, maybe one, they are taking some or in some sample treatments. So we can have single specific or signals detected to only 1, 2, 3 volunteers, so they are not representative. So they should be another exclusion criteria. Another criteria that you can use, or they are frequently used, is the quality related to the QCs. You analyze multiple QCs through all your sequence, although you can perform some normalization steps to try to normalize these analytical drifts. There are still features that they were not able to normalize properly, because they were impossible to using different algorithms, or they were the run was. So maybe another criteria is use the coefficient of variation or relative standard deviation based on the QC. Depending on the study, people using below 20%, below 30%, depending the street you want to be, you can filter it. And also, as I say, in this type of study, in acute intervention study, so we have the placebo samples. So as I say, if we have seniors detected in the placebo samples, we are looking for metabolites coming from the strats. So for that, we use the placebo to use them later for a filtering. So for that, we consider also filtering based on the placebo sample, also based on the time zero sample before the intake of every single volunteer. So we have many potential filtering criteria. Is decide them on the design of your study, and based on the objective and the design of the study, define the criteria and filter it. But you see, there are criteria that they can be common for every single study, like based on the blanks in the QCs. Another, they can be specific of the study.
So the next question, how many QCs usually use in your protocols analysis? Use internal standard or isn't necessary in the batch?
So how many QCs? So basically, I put in the slide some of the typical sequence that we usually run. So at the beginning, we usually run like between 5 to 10 QCs consecutively, especially to equilibrate the system. And then we usually run through between every 6 to 8 samples, depending on the study. But we run also depending on the method. If we're using a chromatography method that is longer or shorter, so depending, but usually between 6 to 8 samples, every single to 8 samples, real samples, we put a QC. And as I say, I don't usually use internal standard. There are, especially the internal standard. So in my case, for example, we use a reference calibration standard, but the objective of that is not like a general internal standard. It's only to calibrate the mass accuracy through the sequence. But this is because of the design of the Agilent instruments. So there are two masses that are continuously injected in the MS, but usually to correct the mass or to correct the mass accuracy every single scan. And also we can use, for example, standards to perform an SST before the runs to check, okay, you can have a mixed standards to, with these 10 standards, you can run it, and you can run like a single run before the real sequence, only for pre-check if the instrument is running well. If you can detect them, the retention times are correct. So for that, I would recommend them, for example, to use some standards in the system suitability test. But internal standard as the main concept of internal standard in your real samples, in Untargeted Metabolomics, based on the LCMS, maybe it's not frequently used, but I know that, for example, I am also part of the NQAC consortium, the Mass Quality Assurance and Quality Control Consortium, and now there are a specific discussion about that. It's something that hopefully it will be great. You can have a mixed standard that can be used for untargeted metabolomics. I know that, for example, for GCMS is more frequently used, but especially to check the efficiency of the derivatization reactions.
The next question is, when identifying a compound using a standard and retention time, what difference threshold do you use?
It's so basically, if you run your real sample, and after your real sample, you run the samples in which you were able to annotate that compounds, and in the consecutive position, you run the analytical standard, the threshold should be as low as possible, because if you have a retention time drift in two consecutive samples, is that something is not running well in your instrument, or is not the compound the compound that you present in the that you are able to identify in your sample. So my advice is you have strong evidence that you have this metabolite in your sample. You buy the analytical standard, then prepare again your sample in which you analyze that compound and analyze together in the consecutive way the analytical standard, and you should see identical retention time and identical MS or MSMS fragmentation spectra. You see different retention time, although you can see 0.2 minutes or 0.1, be careful.
So the next one, how to purify the compounds for the clinical trial after their detection using an MES instrument.
So basically that is an important question if I understood well. So it's like I understood that we, for example, we identify some specific markers or some of these metabolites that we were able to identify in the biological samples, and then we need to move or to try to run some in vitro, in vivo, or clinical assays with this specific compound. So that is an important question. First of all, I would like to try to acquire or if they are available in the commercial sources, if it's possible to buy it, it's going to be the easy way. If not, it's going to be complex because we need to see, because also for a clinical trial, you need a lot of amount. It's not, so you can isolate them if you want to do a test, like for example, or you want to elucidate, like for example, perform an NMR analysis to elucidate better the structure. Then maybe you can use preparative LC system, but to perform a clinical assay is very, very challenging. Also because if you want to use later for giving them to people, you need to also to take into consideration safety regulations to if you are going to have the permission to use them for the clinical study. I don't have experience with that, so we are not performing clinical trials with isolated compounds. So as I said before, the longitudinal assays that we are performing is with olive strats instead of isolated compounds.
So the next one, how do you usually analyze MES spectra to identify the neutral mass and fragments of a specific compound? For instance, if you have 250 samples, how do you determine the representative MES spectrum?
Let's see if I understood well. So basically, so first of all, so one of the best samples in which you can perform some MSMS analysis, if you prepare the pure QC, they're going to be the pure QC because basically they are covering the average levels of all compounds for every in one single sample, right? But the problem is, for example, some compounds, for example, in this type of study, we perform some DDA analysis in the QCs. Some of the important compounds that we discovered, they were not present in them because maybe, as you see in the course, maybe in the olive, they were only present at time after 0.5 hours, 1 hour, 2 hours, and then they were diluted. So when we prepare the QC, the metabolite in the QC were not enough concentrated to be able to detect. So for that, I would recommend to try to optimize because it's something that I was able to optimize last year in my lab, is try to optimize DDA method with a very good frequencies in the combination of MS1 scans and MSMS scans, which allows you to fragment it to analyze all your samples in DDA method. But you need to optimize very, very, very well. You need to, because you need especially have enough number of scans. Also optimize very well to don't lose sensitivity in the instrument. And of course, have the opportunity to obtain. So my main advice is to try to optimize a DDA method. I saw that it was possible. The result that I present today, it was from years ago. The acquisition, and it was not using that method. In the second that we run in this sample, basically the MSMS analysis were done in the QC. But as I saw you later, then we detected, okay, some of the analytes that I want to annotate, I don't have the fragmentation spectra. And then what do we do? What did we do? So basically we went again to take the samples, and then to the samples in which we have the evidence that these compounds were present, and then perform like a target MSMS analysis to these samples, to specific metabolites in which they were significant.
So another question. When dealing with plant metabolites which belong to various categories, can be extract sample with different strategy and pull in the end, dry and reconstitute to get more number of compounds annotated. Would it be useful?
I think it's a good idea. So, but the point is like to report later the way, or I think it's not. It sounds. I think sounds to me. I never thought about that, but I think sounds reasonable to do that, because you can perform different extraction, then pull them, and then try to analyze the pooled based on different extraction methods that they can cover different families to only run one sample instead of multiple ones. But the point, maybe one of the drawbacks of that is like it's going to be difficult later to report some or to link which a methodology, extraction methodology is better for specific compounds related to your sample. But you don't have enough number of budget to run multiple samples in your LCMS system, and you don't want to see the difference between the metabolites present in different solvent extraction methods. I think it's a good idea for optimization of the resources of the lab, of the time, and to get a first impression of all the compounds. And I think it's a very interesting idea. I will think about that in the future.
Yeah, but I have like a comment on that from my own experience working with multi-phytochemical from different chemical families. The thing is that maybe you can extract them with different type of solvents, but you will need different chromatographic methods for them, because polar compounds, they need to be analyzed with a Helix method. Non-polar compounds, they can be run in a reverse phase column. So maybe you have to check that if all of these type of compounds can be analyzed in the same chromatographic method. Maybe that would be a point to optimize.
That is an important point. Absolutely. You need to consider if, of course, as Daniela said, if you have completely different polarities, you need to consider also to run different chromatography methods. And also another point is like also at the end, also you combined all the strats coming from different that I think it can be possible to do it. But at the end, if for example you dry it, at the end you need to also to choose only one solvent to reconstitute it. And then if you choose, for example, a polar solvent or an unpolar solvent, maybe you are not able to reconstitute all the mixture of compounds that you were able to dry it in the mixture. So as I said, it sounds interesting, but you need to consider the differences in the metabolites you want. If there are very strong difference in the polarities of the compounds, it's going to be very, very difficult to put all together and run in a single analysis.
So one more question. If we are used to work with targeted analysis, how would you recommend to start learning metabolomics, softwares, design, etc., considering that we have the possibilities in the lab instrumentation? Thank you.
So for targeted analysis, I think one of my main advice is try to learn about the analytical chemistry, how to validate an analytical method for quantification, how to report the results, how to validate, how to determine the limit of detection, limit of quantification, how to see if there are matrix effects or not. It's some complex. There are some guidelines that you can see in the, for example, EMA, the European Medical Agency or the FDA, you can see the guidelines and you can see that there are many you want to have a targeted validated method to analyze or to quantify some specific metabolite. You need to perform many tests to validate it. So I would recommend to learn a lot about the tests that you need to do for validation of the targeted method for quantification.
Yes, so now we have some comments about the presentation. Dr. Claudine Manache said, very nice presentation. Looking forward to see the results of the EcoBioLive project at the next conference.
I
Good
hope
luck.
so. I promise.
And also one more question now. I mean that we are already known about targeted, how to start with untargeted metabolomics.
Okay. So maybe I misunderstood the previous question. So for untargeted, so there are many things that you need to get familiar. As I tried to put some of this in this presentation, like get familiar with, of course, with the software, but also with the data pre-processing steps. I can imagine if you get familiar with targeted methods. So, for example, the sample preparation and the chromatography or the mass spectrometry part, you should are familiar. So maybe then I will focus more on data processing software, understand very well the steps, the filtering, the wall of the QCs and all the QC metrics that you need to implement in your analysis to have good quality results. And also get familiar also with the annotation tools, the databases, the optimizing the data dependent or acquisition methods, so data independent acquisition method, depending on your preference, or also get familiar with all the databases, tools, GMPS, Sirius. There are many. So as I say, or one of the messages that I want to say in this webinar is that there are very good free open source software to get familiar and also to watch tutorials, videos. And if you have any specific question or you need any specific advice, or write me an email, everyone that you are attending this webinar, I will be super happy and to answer you and help you.
Thank you, Álvaro. We have no more questions. I will pass the word to Daniela.
Yeah, I'm really, on behalf of the EMN, I think we are all really grateful for your presentation, on your predisposition, for answering all these questions in such a detailed way. So I think we can conclude now this session, this webinar session. Just remember to follow us in our social media communities. For those who were wondering, we are going to upload this recording on the Metabolomics Society website in a few days. So just keep track on that. Well, thank you very much, everyone, for attending this webinar series. Hope to see you in our next webinar session in December. We are going to post the day and the time of that presentation in our social media, so keep track on that. So, well, thank you, Diana, for co-hosting, and thank you, Álvaro, again. We're going to finish now.
Thank you very much.
Thank you, Álvaro. Great
Thank you.
presentation. Thank
Very
you.
good success to all your activities of the EMN. I hope to see you in Prague and enjoy the EMN activities in the next conference. So see you and thank you very much.
See you. Thank you, Álvaro. Thank you. Bye. Bye, Daniela.
Bye-bye.
Bye.
Bye.
Bye. Thank you.